Whoa, hi everybody. This is the walkthrough video for Wednesday. It's December 3rd and try to week 4. If you forgot to or you didn't have your binder, please skip all of yesterday's yellow bell work. There was an assignment yesterday. It's sitting in Canvas waiting for you. You will have a hard time doing it, I think, unless you watch yesterday's video carefully where I do some explanation. And we're also going to work on it today. So here is what? No, we went backwards. There we go. So here's your do now for today. It says an experiment blanks the independent variable and blanks the dependent variable. So go ahead and copy those, pause the video. Okay, we're going to do the second one first. The dependent variable is what you measure. So like if you're trying to test the input of different, like the effect of different colors of lights on corn seedlings, the growth is probably what you'd be measuring. So you'd be measuring growth in centimeters after day 6 or something, whatever it is. It's measures is the second blank. And then the first one is changes. In the example of that corn assignment, it would be the different colors of light, right? So an experiment changes the independent variable and measures the dependent variable. So today we're going to design an experiment about human performance. We started class today with a pre-lab. There's some notes to take, which I'll share with you here. And then people did a quick revisions check on their modeling body systems assignment. We have a new table of contents entry for you today. I'm going to make sure that you and I can both see it. It's the human performance pre-lab. And you know what? We are not going to turn it in. You're going to use it pretty well, but we are not going to turn it in. So I've changed my mind about that from first period. Human performance pre-lab. Once you have it in your hands, you're ready to do this next part. So we've been learning a lot about human body systems, right? And thinking about how they interact. One thing we're going to talk about is what happens when something changes in your body. So homeostasis is keeping your body alive in changing conditions, right? So let's say that your body is moving along as happens all the time, there's an imbalance, right? So there's something out there in the outside world that causes this imbalance and produces a change in this variable. That change is detected by a receptor. For example, your body temperature is regulated by the hypothalamus in your brain. That receptor is a sensor and it sends information or input to a control center. That control center, like the hypothalamus in the brain, decides to do an output. That output is the effector, and that helps you return back to being in balance. This type of feedback is a negative feedback cycle, and negative feedback is not all bad, as we'll learn about today. So the story for today is that you've been hired by the U.S. Olympic and Paralympic teams, which you might think of as Team USA. Right now, they're gearing up for the 2026 Winter Olympics. I told you about our student athlete here at South who went to the Paralympic, like the Para World Games. That's the organizing body. If the U.S. is sending you as a team member, it's this thing. So in our little project over the next couple of days, you've been hired as a kinesiology researcher. Now, kinesiology is the study of human athletic performance, both for athletes and for everyday people, whether you're very active or not. It's studying the anatomy, physiology, and mechanics of body movement. Team USA wants your research group to investigate the effects of different training on the human body, including types of exercise, duration, which means the length of time of exercise, and the intensity of exercise. Each of those could be an independent variable. You're going to choose one. Because you've been studying the human body, you know that many body systems work together to maintain homeostasis through creating equilibrium, which is a steady state, a stable state, or a balanced state, where any changes are canceled through other changes. So now we're going to learn about how, which is feedback, right? So where are we going? Thinking about how these variables end up changing. So we're going to take some quick notes. This is in that document. You'll see that these particular graphs are here, and there's a place to do examples. So importantly, negative feedback isn't bad. It helps us keep a set point, right? So here's a classic example of what a negative feedback graph looks like. The numbers get big, something happens in the system, the numbers get small, something happens, the numbers get big, right? That set point is like a thermostat in your house, right? If it's cold outside like it is now, the heater heats up your house, it turns off, it gets cold in your house. Eventually the heater turns on and your apartment gets warm again, and then it cools off. And it turns out the same thing is happening in your body. So let's say that this is where you want your body temperature to be, somewhere around 98. This increase in body temperature, it means that the hypothalamus detects that increase in temperature, and then you end up with effectors like sweating, like your blood vessels dilating to allow more blood out to your extremities because they cool off faster, and the hairs lie flat, right? Now, let's say it gets too cold, and that means that your body closes the blood vessels in your hands. Closes the blood vessels in your hands and arms to bring that back into you. And then your hair rises, right? So the classic example is thermostats in buildings, but also human body temperature thermostats. Now, in contrast, positive feedback is for things that gotta happen fast. I'm gonna say gotta happen fast. They have to happen quickly or they're gonna be a big change. And the graph looks like this. It looks a little like a dip here. It should just be a parabola. And then something big happens at the end. And our classic examples are blood clots and babies who are born without surgery. There's a feedback loop about baby pushing on the cervix getting bigger through a chemical mediator, and baby therefore being allowed to settle harder. So all of a sudden, there's a big change. To think about blood clotting, that's mediated by platelets. They are small particles in your bloodstream. And when one of them shows up to a site, they recruit more and they recruit more and they recruit more. And then you can see the wound in your capillary or blood vessel is patched. Your body, it can be a tube again to allow your blood to not spill into other areas in your body. So classic examples. So now we've described the idea and effects of feedback mechanisms in general. Now you're going to explore some phenomena in which feedback maintain homeostasis. We're going to think about exercise for that. And we're going to identify what data we need to collect to answer a research question and why it will serve as relevant evidence. So the stimulus or stimuli that makes changes can be either biotic or abiotic. And you're going to write a little definition. Biotic is living, like a living systems thing, and this is not alive. So you just pause the video and write those down. Now, this is going to be important information for you as we design an experiment. You're not here to design it, but you do need to be able to design experiments, so listen carefully. Team USA athletic trainers are asking you to design an experiment to investigate a change in exercise training on young people on the circulatory and respiratory systems. Now, this is important. As people do more intense physical activity, they need to increase the amount of oxygen to their muscles and they need to breathe out CO2, right? And to do that, the reason you're breathing it out through your lungs, through your mouth and your nose, is that you are removing it from your bloodstream. Why? We'll talk more about that in Unit 2. But so we thought about what would be easy to count about circulatory and respiratory system. We thought about heart rate as a dependent variable and breathing rate. And it turns out we're going to measure both because they're both interesting. So this one has two dependent variables. So what we did today was think about what it was you were going to change. Remember, there are a couple options here. They're all the way back here. My apologies. There. Type of exercise, duration or length of time of exercise, and intensity of exercise. And so folks went through and filled out this diagram. And then they thought about controls. Like one of the things we wrote here was that because different people have different exercise habits and because exercising a lot decreases your heart rate, it makes it so your heart rate doesn't go as high when you're doing vigorous exercise or moderate exercise as somebody who has been practicing a lot of athletics. We're going to say for that control variable, it has to be the same person. Another control variable is the same day. People who are student athletes know that some days you wake up and you feel great, you've really got it in you. And other days you wake up and you just don't got it. So having it on the same day means that your exerciser, they don't need to be a student athlete or even an active student, will help make it so that they can be successful. So no need for an instructor's okay. And we talked a little bit about the procedure. Now, before class ends, your colleagues went and they took a look at your body systems project. Remember this checklist that has that has these things in it. They went through and did a first assessment about whether their thing has that thing or not yet. And I'm going to put them on the wall in the next week or so, and that'll help us make sure they're all corrected before the 6-week deadline. All right, so you're going to write this sentence down. You're going to look at the graph. You're going to remember what type of feedback it is. You're going to write that word on the blank. All right. I wish you were here. I hope things are okay with you. We'll see you soon. Bye.